Bismillahirrahmanirrahim. Assalamu alaikum. Today we are going to discuss autoimmunity and autoimmune diseases. Learning objectives for this lecture are to know about the autoimmunity and self-tolerance, then what are the autoimmune diseases and its classification, basic mechanism of the autoimmunity and its diagnosis, and then finally some important autoimmune diseases such as systemic lupus erythematosus, rheumatoid arthritis, and SCID. Antigen is a substance that is recognized by our immune system as a foreign, and as a result, the immune response is elicited against it. We also know that antigens can be self or foreign. The foreign antigens are not part of our body, such as the bacteria, viruses, pollens, etc. But the self-antigens, they are the components of our body, for example, our own body cell surface molecules and proteins. The main function of the immune system is to provide protection against the invasion of these foreign antigens. Our immune system has the ability to react with enormous and diverse foreign antigens while not reacting or mounting immune response to self-antigens. So this means that our immune system takes and tolerates their self-antigens. Now, if the immune system starts responding to our self-antigen, it will lead to autoimmunity. So the autoimmunity can be defined as induction of an immune response against one's own tissue because of the breakdown of mechanisms which are responsible for self-tolerance. So this means that the immune systems will start attacking itself. Further, this may result in an autoimmune disease in which individuals' own cells, tissues, and organs, they are damaged as a result of the immune system attack. So let's see what is self-tolerance. Self-tolerance refers to the lack of responsiveness of immune system to the self-antigens. Self-tolerance enables our cells and tissues to live in harmony with an army of the lymphocyte. And it is achieved and maintained by the central and peripheral tolerance mechanism, and we are not going to discuss it here. Therefore, the autoimmune diseases are those disorders which are caused by the autoimmunity. Around 1 to 2% of the general population is affected by these autoimmune diseases. Now, coming towards the classification of autoimmune diseases, autoimmune diseases are both organ-specific and non-organ specific. In these, both the cellular and humoral immunity are involved. It is still not certain why or how the autoimmune disorder occur, but the contributing factors including genetics and the environmental conditions that can trigger the disorder. Some of the known environmental triggers can include the drugs, viruses, bacteria, food, pollutants, etc. The combined effect of these factors, they can lead to the dysregulation of immune system, and thus the autoreactive cells will be activated, and they will start destroying the self molecules or antigen, and thus the autoimmunity will be ensued. Autoimmune diseases are diagnosed on the basis of symptoms and detection of the autoantibodies. The antibodies which are directed against cells or tissue associated antigens, they are detected by the indirect immunofluorescence, and antibodies which are directed against the soluble antigens present in the circulation, they are detected by ELISA or radioimmunoassay. Now coming towards the systemic lupus erythematosus, this is actually a prototype of a multi-system disease of autoimmune origin. and it is characterized by the autoantibodies, particularly anti-nuclear antibodies. Systemic lupus erythematosus is a chronic, remitting, and a relapsing disease which is caused by injury to the skin, joints, kidney, and serosal membranes. Antibodies in the SLE will be directed against the self tissues or cells, and thus they will mistakenly recognize themselves as the self antigens as the foreign antigens. It is still not known what causes this disease to start, but one thing is for sure that it is due to failure of mechanism that maintain the self-tolerance. Antibodies have been identified against an array of the nuclear and cytoplasmic component of the cell that are neither organ or nor species specific. In addition, a third group of the antibodies are directed against cell surface antigen of the RBCs. Now, pathogenesis of systemic lupus erythematosus in genetically susceptible individuals, the genes encoding class 2 MHC molecule and the complement system proteins, along with some environmental antigens or proteins, they can lead to activation of the helper T cells and B cells, which are normally specific for the self antigens. So, as somehow the body's immune system does not recognize these self antigens, and as a result, the B cells they will start producing antibodies against their own cells, and these antibodies are the IgG antibodies. So, as a result, the self reactive antibodies and the immune complexes they will start accumulating in the tissues and they will start damaging them. All these effects are evident as clinical manifestation. Females are more prone to get SLE. So, SLE is a multi-organ disease affecting skin, joints, kidney, heart, etc. The most important clinical manifestations are the butterfly skin rash, photosensitivity, then nephritis, pericarditis, etc. The laboratory studies of SLE include positive anti-nuclear antibody, positive double-stranded DNA antibodies, or these are also called as the positive lupus erythematosus antibodies, then the positive anti-SM or anti-smooth muscle antibody, elevated CRP, ESR, but decrease C3 and C4 serum complement proteins. Immunofluorescence pattern for SLE includes the peripheral rim, but it could be diffuse, speckled, or even nucleolar. But the peripheral rim pattern is more specific for detection of the autoantibodies in case of SLE. Now, rheumatoid arthritis, it is another autoimmune disorder which particularly affects the joints, lacrimal glands, salivary glands, and lung, etc. The most striking clinical manifestation is the in severe cases of rheumatoid arthritis, they include the swan neck deformities of hand fingers and thumb. Now let's see the mechanism of the autoimmune disorder. The helper T cell, they will release interferon gamma and IL-17. The interferon gamma will activate the macrophages and IL-17 will recruit the neutrophils. So in turn, these activated macrophages will start releasing tumor necrosis factor and IL-1, which will then in turn triggers the release of proteases. And the activated T cell, they will express rank L ligand, which will then promote bone resorption. Lab finding for the rheumatoid arthritis include the positive rheumatoid factor, and RF is meant by the IgM antibody produced against IgG. But this RF factor is not specific for the rheumatoid arthritis. Then there is a new antibody test which is called as the anti-CCP or antibody to cyclic citrullinated peptide. This is more specific for the rheumatoid arthritis. Now coming towards the severe combined immunodeficiency syndrome, which is also called as the SCID. This is actually a group of the rare disorders and which is caused by mutations in different genes which are involved in the development and function of the infection fighting immune cells. The infants with the SCID will appear healthy at birth, but they are highly susceptible to infections. So if left untreated, these infections will start or will result in a fatal outcome in first years of the life. Defects occurs at the common lymphoid progenitor or the CLP stage, or those affecting processes that are common to the B and T cell development. They all combinedly they result in the SCID. So the multiple genes such as ADA or JAK3 mutations, they are usually seen in SCID patients. The clinical features of the SCID patient, these include the recurrent infection, oral candidiasis presenting with a prominent thrush, chronic diarrhea and failure to thrive, extensive rash, and morbilliform rash due to the reaction of maternal T cells that are directed against the fetus, which will then later on develop into graft versus host disease. Diagnosis of SCID is basically it is based on the clinical manifestation, and usually it is said that SCID, a diagnosis of the SCID should be suspected in children with any of the following condition, such as the unexplained lymphopenia, failure to thrive, chronic diarrhea, recurrent severe episodes of infection with the human herpes virus, varicella zoster virus, measles, influenza, parainfluenza, etc. And in addition to this, a family history of SCID is also very prominent and it should be considered. That is all for today. Thank you very much.